Hello, welcome to another video. And this time I found this article about genetics and schizophrenia, and I want to share this with you. The title is a genomic lifespan program that reorganizes the young adult brain is targeted in schizophrenia, and to understand in depth we should read the abstract of it. The genetic mechanisms regulating the brain and behavior across the lifespan are poorly understood. Obviously poorly understood, and we found that recently genetic studies in schizophrenia are quite popular, and most of the studies of schizophrenia and genetics are filling the journals. We found that lifespan transcriptome trajectories describe a calendar of gene regulatory events in the brain of humans and mice. Transcriptome trajectories define a sequence of gene expression changes in neuronal, glial, and endothelial cell types, which enabled prediction of age from tissue samples. A major lifespan landmark was a peak change in trajectories occurring in humans at 26 years and in mice at 5 months of age. That's interesting. This humans and mice conserved peak was delayed in females and marked a reorganization of expression of synaptic and schizophrenia susceptibility genes. The lifespan calendar predicted the characteristic age of onset in young adults and sex differences in schizophrenia. We propose a genomic program generates a lifespan calendar of gene regulation that times age-dependent molecular organization of the brain and mutations that interrupt the program in young adult cause schizophrenia. So here, because they focus on catching the time of having schizophrenia. I'm not sure what is their field, and the thing is, it's not easy to catch the first age of onset, and because we assume that when they go to psychiatrists they are sick at that time, but actually it's earlier probably, and actually I think it's slowly progress, and that's why we cannot really be sure that when is their first psychotic symptoms have been appeared in their lifespan. But it is interesting, and people, we should attempt to discover and explore it more in depth. In our lifetime we go through many changes physically and also intellectually. So this is the comment of the journal. We are particularly vulnerable to develop psychiatric disorders, and the majority of mental conditions start to manifest in teenagers and young adults. The symptoms for schizophrenia, for example, a mental health disorder in which patients often experience hallucinations, delusion, or change in behavior, typically start in the mid-20s. Schizophrenia tends to run in families, and it's likely that different combinations of faulty genes that affect the connections between nerve cells increase the chance of having the disease. Until now, scientists have assumed that certain situations and environmental factors trigger the condition, but it was unknown if genes could influence the age at which disease will begin. To explore whether genes in the brain change at certain time points, this author and his colleagues examined how the genes are turned on and off across the lifespan of healthy mice and humans. The results showed that in both mice and humans, a genetic lifespan calendar controlled every cell type in the brain and directed the way they worked at different ages. The timing was so precise that It was possible to tell the age of a mouse or a person simply by looking at the way the genes were expressed in a tissue sample. That is interesting. So that they're mentioning this study for 2 paragraphs, and the same group studied how the genetic lifespan calendar controlled the genes damaged in schizophrenia and found that the calendar caused a major reorganization of the genes at a time when the symptoms started. This suggests that the genetic lifespan calendar is a crucial factor that can determine at what age the disease will start. The next step will be to study how the genetic lifespan calendar programs change throughout the brain and explore if it could be manipulated to change how the brain ages. This could help to develop new types of treatments for schizophrenia and other conditions of the brain. So that's interesting, and let's see. Gene expression trajectories can be classified and quantified based on the characteristics of their turning points. And quantifying expression trajectories. Which measured mRNA expression levels from 269 prefrontal cortex samples across the human lifespan. Although TTTPs were identified across the lifespan, they were sharply concentrated during early adulthood, and summing the number at each age shows a striking peak and mean of 26 years in males and 27.5 in females. These data indicate that despite greatly different lifespans, these two mammalian species share a lifespan program of brain gene expression with conserved features. The complex trajectories were found throughout lifespan, suggesting they could be used to predict the biological age of the brain. To test this, we used radial-basis support vector machines and demonstrated that classifiers trained on partitioned subsets of the gene expression data predicted age in the test sets with an accuracy in humans of 5.5 years and 28 days in mice. That is interesting. This indicates the genetic lifespan calendar of transcriptome events is a conserved feature of mammals. So this article seems a very long one, and so these are the revisions. The text needs attention. The introduction is long and meandering and will be more accessible if shortened significantly. The authors need to explain what they have done with greater clarity. For example, the model the authors propose is so unclear and meaningless. Whoa, that is harsh. Nearly meaningless. But just saying nearly, maybe it's not that harsh. We acknowledge that the text will benefit from improvement in clarity and brevity. We have edited the abstractness significantly shortened. So this is the answer, the introduction and discussion. And this is the short form. Actually, it's quite long. And we have also rewritten the model and introduced the praised genetic lifespan calendar to help convey that the gene regulatory events occur particular age. The authors together which models they used and why. There are several algorithms presented in this work, of which only TTTP is straightforward. It's hard to understand why the authors use one over the other. So they explained it. Comparison of inner units across space is also unclear. Is a human year equivalent to a mouse day? We inadvertently had left these details out of the methods, and thank the reviewers for bringing it our attention. We have amended the materials and methods section to make this clear. Correction for multiple testing needs to be made explicit. For the self-test analysis, a confirming correction is used, but it is not clear that appropriate corrections were made for other analyses. That is interesting. We have made the use of multiple testing correction more explicit in the paper. P-value is Bonferroni corrected. We now denote it as where a p-value is Benjamin Holberg corrected with PDR equals to 0.5. Now denote it as. The materials and methods section now makes clear for every analysis performed whether and how multiple testing correction was performed. Okay, it seems they asked 5 questions, 6 questions, 8 questions. In total, it seems the communication seems to last like very long time. And thanks for watching this video. And if you enjoy article, you can find it online, and I will describe the link in the description of this video. I wish you have a beautiful day.